My name is Maria Clara Ohno, and I'm a student at the Brazilian Swiss School of Curitiba. My project, titled Characterization of Neurospheres Derived from Stem Cells from the Dental Pulp and the Apical Papilla, was conducted under the advisory of Professor Dr. Letícia Fraccaro and Carmen Rebelato. Mesenchymal stem cells are multipotent and self-renewing. When sourced from the apical papilla and dental pulp, they can form neurospheres in 3D cultures in the presence of neurotrophic factors due to their neural crest origin. Their enhanced neurogenic potential and the in vivo-like conditions of 3D cultures make them promising for treating and modeling neurodegenerative diseases such as Parkinson's. The main objective of this study was to characterize neurospheres derived from DPSC and SCAP during the process of neuronal induction for neuronal differentiation, while the specific objectives were to thaw and culture SCAP and DPSC, induce the formation of neurospheres from DPSC and SCAP, characterize the neurospheres, and compare the results obtained. Following PUC-Paraná Ethics Committee's approval, 2 samples of SCAP and DPSC were isolated, expanded, cryopreserved, and eventually thawed for culture. Viability was assessed, and cells were transferred to non-adherent well plates with DMEMF12 with 15% FBS. Neuronal factors, including B27, EGF, FGF, and PEN-STRAP, were added only in the differentiation group. 3D cultures were maintained for 5 days, photographed on days 2 and 5, and neurospheres were quantified, measured, classified by size, and analyzed by ANOVA. Results show that after thawing and culturing, nearly all samples exhibited viabilities greater than 80%. All samples adhered to plastic and had fibroblastic morphology. No significant differences were observed between DPSCs and SCAP in neurosphere number or size on days 2 and 5. From day 2 to 5, both SCAP diff and DPSC diff showed a significant reduction in neurosphere quantity. Size distribution on day 5 was similar across groups except DPSC control, which increased in small neurospheres. Large neurospheres did not differ, but medium ones significantly decreased in DPSC control, DPSC diff, and SCAP diff. Nevertheless, the concentration of medium neurospheres throughout the cultures predominated relative to all other sizes. The high viability confirmed an effective freezing and thawing protocol. Cells also met the International Society of Cell and Gene Therapy criteria, confirming their identity as mesenchymal stem cells. Finally, in analyzing neurosphere formation, the addition of neuronal factors to 3D culture may have exerted selective metabolic pressure on DPSC and SCAP, allowing resistant subpopulations with higher nestin expression to persist. This expression maintains cells in a proliferative, undifferentiated state, supporting self-renewal and neurosphere formation. According to Gonmani's study, large neurospheres show varied nestin expression across layers, while medium ones display strong homogeneous expression. Thus, large neurospheres may drive uncoordinated self-renewal and neuronal potential among cells. On the other hand, medium-sized spheroids are more stable, likely reforming after fragmentation as resistant cells aggregate into spheroids around 200 microns to promote the continued expression of neural stemness markers. This size is metabolically favorable, ensuring efficient nutrient diffusion, survival, and coordinated neuronal differentiation. Cell adhesion and extracellular matrix deposition increase compactness, preventing growth beyond 250 microns and maintaining most neurospheres under 200 microns. In conclusion, DPSC and SCAP show comparable neurosphere formation in 3D culture, likely reflecting their common embryonic origin. Furthermore, the predominance of medium neurospheres may relate to marker expression favoring neuronal differentiation, supporting 3D culture protocols as promising for generating neuronal phenotypes from mesenchymal stem cells.